Hello and welcome everyone to the LEXOGEN webinar. I'm Ekaterina Alexeyeva, a Senior Marketing Manager at LEXOGEN, and I will be moderating this webinar today. Before we start, I have some announcements. Please note that all attendees are in listen-only mode. Our webinar today is about LUTHOR, unparalleled sensitivity in single-cell sequencing. Our speaker is Dr. Yvonne Goepel, the Product Manager at LEXOGEN. This webinar will take about 55 minutes, including 40 minutes of a presentation and 15 minutes of the Q&A. For the Q&A, we invite you to submit your questions by typing them in the chat section, which you can find on the control panel of the webinar software. Feel free to type in your questions during or after the talk. However, please be aware we will only answer them after the talk, during the actual Q&A session. If you have any technical difficulties, please let us know about it also in the webinar chat box. And now it's my great pleasure to turn it over to Yvonne. Welcome and please go ahead.
Thank you, Ekaterina. And welcome also from my side. Before we start with LUTHOR, I'd like to take a few minutes to introduce our company to you. After that, we will take a deep dive into the Thor amplification technology and LUTHOR 3' mRNA-Seq. After that, we will come to performance data and applications for LUTHOR for ultra-low input RNA and single cells. And in the end, I will hand back to Ekaterina, who will lead through the question and answer session. With that, let's begin. LEXOGEN is a biotech company that was founded in Vienna, Austria in 2007. It focuses on developing technologies for complete transcriptome sequencing and RNA and everything RNA. The US branch was founded in 2014 to support sales and tech support within the US. All R&D, services, and manufacturing is located in Vienna, Austria, providing a sustainable local company culture with an interdisciplinary, multinational team of RNA experts. LEXOGEN also employs a worldwide distributor network to offer our products, technical support, and services to customers all around the world. LEXOGEN is a complete solution provider. We offer full sample to sequencing service, including data analysis, for highest convenience in RNA-Seq, consultation and expert technical support, and a variety of products and data analysis solutions for RNA sequencing. Our full service also includes sample preparation, RNA extraction and pre-processing, library preparation, sequencing and data analysis. Service is available for all our products, including gene expression profiling using 3' mRNA-Seq. We also offer whole transcriptome sequencing in form of total RNA-Seq in combination with rRNA depletion or as whole transcriptome mRNA sequencing. LEXOGEN also provides small RNA-Seq service and time-resolved gene expression profiling that allows to identify nascent transcripts by metabolic labeling and can be used to assess transcript kinetics and turnover. Our expert technical support is available to every customer and can be used throughout the complete process. LEXOGEN employs application and field application scientists, including specialists for bioinformatics, and offers help and support at every step of the way. Before you even start, tech support is available to help you plan your experiments and can consult you on experimental design and aspects to keep an eye on to answer your specific research question. This includes sample type and input used, enrichment strategies, if any, the number of replicates that are recommended, and controls to be included in the Seq experiment, as well as the appropriate library preparation method and multiplexing strategy to use at hand here, and considerations such as sequencing depth and read mode. Before or during sample preparation, you can also turn to us for best practice sample preparation advice or for automation, as well as troubleshooting or even hands-on trainings. While we do not provide sequencing machines ourselves, our kits are developed to be fully compatible with market-leading sequencers. Our tech support team is happy to help out here as well and provide you with information on loading recommendations as well as troubleshooting. And if you are unsure about how to analyze or interpret your sequencing results, we are also offering data analysis solutions and support, including our plug-and-play solutions, and tech support can advise on results, parameters, and settings to optimize your results, and advise on how to best evaluate spiking controls. LEXOGEN's products cover every aspect of an RNA-Seq experiment, from kits to use for sample preparation or customized solutions, over software and data analysis pipelines that are built in-house and offer the highest quality results possible. Here, I just want to show you an overview over LEXOGEN's portfolio, and I know there's a lot going on this slide. But what I want to point out is that all aspects of a complete RNA-Seq workflow are covered within the LEXOGEN product portfolio. And LEXOGEN is a very innovative and fast-paced company, which is also reflected by the new additions to the portfolio. All products marked here are new releases from this year. From the metabolic labeling kits that are applied to living cells and organisms prior to RNA extractions over various RNA extraction kits and pre-processing kits, we cover whole transcriptome sequencing as well as small RNA sequencing, and our 3' QuantSeq product line for highly efficient and easy expression profiling. Non-expert data analysis solutions are offered for QuantSeq, for gene expression profiling, and for Coral for whole transcriptome RNA-Seq. We also offer full-length cDNA kits that can be used to generate cDNA for long-read sequencing and spike-in RNA variant controls, our SERVs, that are used as universal controls for all RNA-Seq platforms. Our 12-nucleotide unique dual indexing systems are the most sophisticated index sets for Illumina sequencing on the market. The newest addition, and the one which we will focus on today, is LUTHOR 3' mRNA-Seq for ultra-low input and single cells. And with no further ado, on to the LUTHOR kits and the novel Thor amplification technology. Conventional single-cell sequencing methods are limited by the low efficiency and conversion rates, and therefore they require analysis of hundreds to thousands of cells whose transcriptomic profiles are averaged out and merged to an assumed gene expression profile. These cells are usually sequenced at very shallow depth, as due to the relatively low conversion rate, only the highly abundant transcripts can be captured efficiently. In contrast, LUTHOR offers unparalleled sensitivity and thus allows an in-depth view of the transcriptomic landscape of one individual cell. Higher sequencing depth can be applied, and even medium and lowly abundant transcripts are captured, so that one cell can be analyzed at high resolution. No merging with other cells is needed, and therefore unique changes to the transcriptome of one cell are not averaged out across a population. LUTHOR thus allows to study the smallest perturbations on a single level. But how is this possible? The key technology of LUTHOR 3' mRNA-Seq is the unique Thor amplification. Thor stands for T7 High-resolution Original RNA and allows the direct amplification of endogenous mRNAs without using a cDNA intermediate as a template. But before we take a closer look to the molecular principles of the method, I'd like to summarize the 4 basic steps of LUTHOR library preparation. The first step is cell lysis. This one is optional in case ultra-low input RNA is used, which is usually purified beforehand. The second step is Thor amplification, which generates RNA copies directly from the endogenous mRNA. In the third step, library generation, These RNA copies are then converted to primary single-stranded cDNA libraries in a single, highly efficient conversion step. The fourth and last step is library amplification. Here, PCR is performed to generate the second strand, amplify the libraries, and complete the adapter sequences for cluster generation on Illumina machines. Also, indices are introduced for multiplexing at this step. Input material for LUTHOR are either intact single cells or already purified RNA. LUTHOR's input range is between 1 to 100 cells, or approximately 10 picograms to 1 nanogram of purified RNA. In case purified RNA is used, the cell lysis steps are skipped and RNA is directly amplified. Cells can be either used directly from suspension and mixed with LUTHOR lysis buffer, or they can be sorted, for example with a fax machine. After cell sorting, for cell sorting, LUTHOR lysis buffer can be prepared and cells can be directly sorted into the buffer. Alternatively, cells can be stored at -80 degrees and LUTHOR libraries can be prepared later when it is more convenient. For best practice, cells should be intact and viable throughout the sorting process until lysis. Cells should therefore be handled quickly and carefully. During sorting, care should be taken to counter-select and remove dead cells and debris. After sorting, cells should be kept on ice or frozen at -80 for storage. This protects the cell itself and also reduces endogenous RNA degradation, as RNases are less active at 4 degrees. After lysis is performed, either on fresh or previously stored cells, RNA can be amplified. Upon lysis, DNA, proteins, and RNA are released, and the RNA is now accessible for amplification. Thor amplification works selectively on polyadenylated RNA and efficiently excludes ribosomal RNA, which makes up the majority of RNA molecules in the cell. In the first step of Thor amplification, an oligo-DT primer is annealed to the poly-A tail of the endogenous mRNA. This oligo-DT primer already contains a partial Illumina-compatible sequence, here shown in blue. And it contains a T7 promoter sequence at the very 5' end. Reverse transcription is performed to generate a cDNA molecule, shown here in gray. The main function of the cDNA strand is to remove secondary structures in the RNA and to stabilize the template mRNA for amplification. The cDNA is not used as a template. In the next step, the poly-A tail is removed, and the T7 promoter sequence at the 3' end that was introduced in the previous step is converted to a double-stranded and thus functional promoter sequence. The resulting molecule is a duplex consisting of the original mRNA molecule, which is now fused to a functional T7 promoter and a stabilizing cDNA strand. The cDNA strand is a non-template strand and it is unwound upon amplification. Thor amplification uses T7 polymerase, which can now initiate at the functional promoter located at the RNA's 3' end. In vitro transcription generates antisense RNA copies that contain the partial Illumina adapter sequence at their 5' end, followed by a poly-U stretch that corresponds to the poly-A homopolymer that was primed in the first step. The copies themselves are lacking the T7 promoter sequence and are therefore not further amplified. Rather, the original RNA is amplified over and over again by subsequently initiated transcription events. The major novelty for Thor amplification is therefore that the original RNA is used as a template instead of a cDNA intermediate that is normally used for conventional single-cell-seq or low-input methods. Using cDNA has some severe drawbacks, One of them is that everything that is missed during the conversion from RNA to cDNA is lost for later analysis. This is usually a major limitation as the efficiency of conversion is quite low, especially with such low inputs. A similar limitation does not apply for LUTHOR. First, because the process of generating the functional promoter fusion is much more efficient. And second, because the cDNA that is generated in LUTHOR mainly has a stabilizing function. And even if a cDNA complementary strand cannot be produced from a given mRNA molecule, this does not necessarily exclude the mRNA from being amplified. Another drawback of conventional methods using cDNA intermediates is that the initially produced cDNA can already contain errors. And as these are then used as template, the errors can be propagated, and in case of PCR, even be exponentially amplified. This can lead to a scenario where an error that is introduced in the very first step can even appear like a real SNP. If this is especially problematic for low abundant transcripts, because here all the reads that are generated in an RNA sequencing experiment could be derived from 1, 2, or 3 cDNA intermediates only, making it much more prominent. For LUTHOR, such systematic errors are avoided by the fact that only the original mRNA carrying the genuine sequence is copied over and over in a linear manner. Therefore, all errors that are introduced in this step are randomly distributed, and none of them will take over the reaction and skew the data towards a prominent variant. Thor amplification finally results in a pool of antisense RNA copies that can reach quite high concentrations. The amplification factor is in the hundreds, and therefore also the original RNA molecule is efficiently diluted out by the copies. Subsequent RNA library generation is done using the amplified RNA as input. LUTHOR libraries are generated in a highly efficient one-step conversion. Random primers containing the partial P5 Illumina sequence, here shown in green, are used in a reverse transcription step to convert the RNA copies into single-stranded cDNA. Library generation uses LEXOGEN's displacement stop technology and random displacement stop primers. These primers prevent strand displacement by reverse transcriptases that initiate further downstream, and thus only the most 5' located primer can generate a functional library. This makes the whole process also absolutely fragmentation-free. The initial Illumina linker, that is shown in blue, and here part of the RNA, is also transcribed back into cDNA during the process and makes up the 3' end of the primary library. Here we see the primary cDNA library in more detail. The single-stranded cDNA now contains both partial adapters, P5 in green and P7 in blue. And the insert sequence again now corresponds directly to the original mRNA sequence and the poly-A homopolymer, which is located at the 3' end of the insert right before the partial P7 adapter. During PCR, first of all, the second strand is generated. Then the Illumina compatible adapters are completed for cluster generation, and indices are introduced at the I5 and I7 position. The libraries are amplified and now ready for sequencing. The image below shows the final structure of a LUTHOR library. Here you can see traces of typical LUTHOR libraries run on a microfluidics device, in this case on a bioanalyzer. Overlaid are libraries generated from 1 mouse embryonic stem cell, 1 HEK cell, or 10 HEK cells. The library size distribution shows that most libraries are around 320 to 360 base pairs in average length. Therefore, the insert size is approximately 150 base pairs, and LUTHOR is thus perfectly suited for sequencing between 50 and 150 nucleotides in single read mode. Paired-end sequencing is not recommended for LUTHOR, as the homopolymer stretch corresponding to the poly A tail of the mRNA is located directly at the beginning of read 2, and therefore the quality would be extremely low and clusters may not be called. Now that we have gone over the molecular details of the LUTHOR chemistry, let's have a look at some data and see how LUTHOR performs with a variety of samples. LUTHOR 3' RNA-Seq enables highly sensitive and consistent gene detection already at low sequencing depth of 1 million reads per sample. LUTHOR libraries were generated from ultra-low input universal human reference RNA, UHRR, where 10 to 100 picograms were used and 4 replicates for each of the inputs. Libraries were sequenced on NextSeq 500 in single read 75 mode, and the detected genes were analyzed for 1 million reads and at a threshold of more than 1 counts per million. For all box plots, the box denotes the interquartile range. This means the first and the third quartile, and the whiskers represent the extremes of the data. As you can see from the box plot, approximately 13,500 genes are detected from 10 picograms of UHRR, and almost 16,000 genes are detected when 100 picograms of UHRR is used. The narrow distribution visualized by the interquartiles also emphasizes the high consistency and reproducibility of LUTHOR preps between the replicates. But how does LUTHOR compare to other conventional methods? We repeated the experiment using a UHR input gradient from 10 to 50 picogram and 2 replicates each, and compared the performance with a conventional low input and single cell kit from a different vendor. This kit uses the conventional workflow of cDNA synthesis, template switching, and second strand generation, PCR amplification, and a DNA library prep on amplified cDNA for NGS library generation. The libraries were then sequenced, and detected genes were again analyzed with the threshold of more than 1 counts per million. LUTHOR consistently detects a higher number of genes for all inputs used than the conventional single-cell low-input method. LUTHOR was also used to generate libraries and assess gene detection rates from 3 different cell types: human HEC293 T cells, mouse embryonic stem cells, and Drosophila S2 cells were used at 8 replicates each. The cells were FACS sorted into LUTHOR buffer. They were frozen and conveniently stored at -80 degrees prior to Thor amplification and library preparation. The libraries were sequenced again on NextSeq 500 in single read mode, and the detected genes again were analyzed for 1 million reads per sample and are shown here at a threshold of more than 1 count per million. For HEC293 T cells, roughly 12,000 genes are detected. For mouse embryonic stem cells, 10,500 genes were detected, and roughly 7,000 genes are detected at a CPM larger 1 for Drosophila S2 cells. This nicely correlates with the cell size, the RNA content, and the transcriptome size of these cells. LUTHOR library preps are very robust. They're highly reproducible and they generate very consistent data. Here, correlation plots for gene counts are shown for replicates from 10 picogram UHR on the left. So these represent technical replicates, or for cell-to-cell correlation for MES cells, representing biological replicates of 2 cells. Correlations are generated at 1 million reads per sample, and orthogonal regression was used to calculate R² in log space, delivering values of 0.9 or higher for both of the examples. LUTHOR generates 3' libraries that focus the reads towards the 3' end of the transcripts, as can be seen from the gene body coverage plots. Reads generated from LUTHOR libraries can be efficiently mapped to exons and 3' UTRs, which we will see in the next slide. In general, the distribution of mapped read to different classes, like exons, introns, intergenic, rRNA, and mitochondrial RNA, is also an excellent way to judge the overall quality of the data and the initial quality of the cells inserted into the library prep. For example, generating libraries from dead or dying cells with pervasive RNA degradation often results in shifts of read assignments. Those are represented by a generally low number of exonic reads, while the intronic and intergenic reads are strongly increased. Also, an increase in reads mapping to mitochondrial transcripts is a good indication of poor cell viability, as mitochondria burst upon cell death, releasing their compounds and RNA content, and this then becomes available for library preps. So how does the read distribution look for data generated with LUTHOR? LUTHOR libraries were generated from 10 picograms UHRR or from different cell types, including human HEC293 T cells, mouse embryonic stem cells, and mouse microglia. Consistently, all of these samples generate high-quality sequencing data. Even for difficult samples, such as microglia, approximately 10% of the reads are mapped to exonic sequences. Additionally, the rRNA is effectively excluded from LUTHOR library preps and ranges between 0.2 and 1.5% only. These inputs here show individual differences in the read distribution that are specific for each sample type or input. So in summary, LUTHOR 3' mRNA-Seq reliably represents the endogenous mRNA composition and efficiently excludes ribosomal RNA even from challenging samples. One of the more challenging sample types are microglia cells. So let's take a more detailed look at LUTHOR data generated from mouse microglia. The analysis of microglia cells is of growing interest in neurological and biomedical research. Microglia have important roles in brain homeostasis and in the disease states. They are known to exert neuroprotective or neurodegenerative functions. However, the analysis of microglia cells and their transcriptomes is very complex and requires special care even from highly skilled researchers. To assay expression profiles of functions of microglia, cells are isolated from brain tissue, which is a tedious process. Special care needs to be taken when working with these samples, and preparation should be carried out as quickly as possible, but also carefully to preserve the highly fragile cells and RNA. After dissection of the brains, the cells are dissociated. And next, a myelin removal step selects against myelin-positive cells so that only cells remain that do not contain any myelin. This fraction can then be immunolabeled and sorted, for instance, by FACS. Alternatively, suspensions enriched for microglia cells can be generated and RNA can be extracted. Preserving the cells throughout the workflow is also extremely important to extract RNA of high quality for further processing. LUTHOR libraries were prepared from microglial RNA and sequenced and analyzed. First, gene detection rates were assessed for ultra-low input microglial RNA using 4 biological replicates. The box plots showed detected genes for microglia analyzed at a threshold larger than 0.1 or 1 counts per million. Again, the box denotes the interquartile ranges. For CPM larger than 1, approximately 12,500 genes were detected from these samples. For the same samples, approximately 14,500 genes could be detected when a threshold of 0.1 counts per million was applied. Marker genes are used to classify subpopulations, classes, or subclasses of various cells. Therefore, we checked also for established microglial marker genes, which were readily detected in LUTHOR datasets. The table on the right shows various common marker genes and the gene counts that can be obtained for them from LUTHOR libraries for 2 different replicates. The biological replicates were also then analyzed in a little bit more detail. Correlation plots shown here on the left show excellent correlation between 2 replicates. The correlation was quantified using orthogonal regression again and determined an R-square of 0.98 in log space. Here, uniquely mapping reads were used. The table on the right shows the number of detected genes for each of the biological replicate. So gene detection at both thresholds is highly consistent when using LUTHOR. Mapping class attribution, as shown in the lower right corner, is also highly consistent between the 2 datasets from biological replicates, again showing the majority of reads is mapping to exons. Therefore, LUTHOR 3' mRNA-Seq generates high-quality data from challenging samples, as exemplified by the excellent replicate correlation and highly reproducible gene detection for ultra-low input and of microglial RNA. Before we wrap up, I would like to show you just 2 more examples for difficult sample types. Other quite challenging sample types that are often assayed by cell sorting in conjunction with RNA-Seq are PBMCs, or peripheral blood mononucleic cells, as well as T cells. These sample types are characterized by very low RNA content, comparably high DNA content. Especially for blood cells, the RNA to DNA ratio is inversed, as these cells contain roughly 10 times more DNA than RNA, while this is typically quite the opposite for eukaryotic cells. This represents a unique challenge to discriminate against the DNA background. As you can deduce from the mapping class attribution for LUTHOR libraries from ultra-low input human PBMCs and RNA derived from mouse T cells, LUTHOR effectively generates libraries from the endogenous RNA and excludes the DNA background. In case genomic DNA regions would have been amplified, the percentage of reads that is mapping to intergenic regions would be much higher. However, here it is quite low, with only 10%. Also, the number of detected genes is quite high for the LUTHOR dataset, with roughly 9,100 genes detected at the CPM of more than 1, from 10 picograms of PBMC RNA, and roughly 13,500 genes detected from approximately 100 picograms of mouse T cell RNA. Previously, leading competitor products detect only about half the number of genes for PBMCs at the same sequencing depth of 1 million reads per sample, and even higher input amounts. PBMCs are very useful sample types, and they can be obtained in a minimally invasive way. They are therefore often used in biomarker assessments, disease and treatment studies, and are of growing interest. A high and robust gene detection is therefore key to successfully unravel developmental and immunological pathways and responses elicited during disease and treatment. Therefore, LUTHOR 3' mRNA-Seq offers an in-depth view of single-cell transcriptomics and is thus perfectly suited to unravel networks and regulatory processes underlying cell heterogeneity, differentiation and development, disease progression on a cellular level, biomarker analysis for disease and treatment, cellular networks, cell responses and immunity, or ultra-low input applications, thus as mRNA assessment from biopsies or exosomes and the likes. To wrap up, we'll go quickly over the benefits of LUTHOR. LUTHOR is highly accurate and sensitive. The novel Thor amplification technology directly amplifies only the original RNA molecule and offers the most comprehensive gene expression profiles from single cells. You can detect roughly 13,000 genes from 10 picograms purified RNA, or up to 15,000 genes from one singularized cell. The template switch ligation and fragmentation-free protocol supports ultra-low input, degradation-prone cells, and even degraded samples. Also, data analysis is straightforward. You can save a lot of time and computing capacity since you're using a 3' library prep, and you can simply count the mapped reads and calculate gene expression. Last but not least, LUTHOR is actually quite cost efficient. When first taking a look at the price, this may seem odd. However, what should be taken into account is that Thor amplification technology allows the direct access to ultra-low input samples. There is no need for RNA extraction, DNase treatment to remove genomic DNA, RNA QC, ribosomal depletion or polyase selection prior to LUTHOR library preparation. The sample can be directly processed, saving money and most of all valuable time when omitting all these pre-processing steps that are normally required for RNA-Seq. This makes LUTHOR also quite attractive for inputs that are on its upper scale, like think around 1 nanogram. You can save all of these pre-processing steps. So in summary, LUTHOR is the first truly comprehensive 3' mRNA-Seq kit for single cells and ultra-low input. It uses the proprietary Thor technology for direct RNA amplification from the endogenous mRNA pool. And with this, LUTHOR achieves unprecedented sensitivity and true representation of the transcriptome of individual cells, without the need to sequence hundreds or thousands of cells and to average out the gene expression profile over a multitude of cells. LUTHOR is a very reliable method, devoid of critical steps like template switch or fragmentation, and thus can also be used for challenging samples. With this, we are at the end of our webinar, and before I hand over to Ekaterina to lead us through the Q&A session, it is my pleasure to announce also our LUTHOR promotion, because what can be better than publishing a paper on in-depth single-cell transcriptome analysis? We would like to celebrate your publication with you. So for the first 5 researchers who publish using the LUTHOR kit, they will get reimbursed for their kit purchase. For details in terms of conditions, please have a look at our website. And with this, I would like to hand over to Ekaterina, and we're looking forward to answering your questions.